Hello and welcome to this Nature Research custom media webcast titled Using Llama VHH to Optimise T-cell mediated immunotherapies. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Isagenica. We'll begin the webcast with a presentation from two scientists at Isagenica. They are Dr. Ed McGowan, Director, Antibody Discovery and Development, and Dr. Mandeep Sehmi, Business Development Manager. We'll then move on to a question and answer session. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them today. And now, over to our speakers.
Thank you, Sarah, for that introduction and for everyone that has joined us today. A quick introduction to the speakers. So, Dr. Ed McGowan is our Director of Antibody Discovery. He trained as an immunologist at the laboratories of Professor Charles Kelly at King's College London before acquiring extensive experience in single-domain antibody discovery and development through progressively more senior roles at Ablynx and Oceanics prior to joining Isagenica. Myself, my name is Dr. Mandeep Sehmi. I trained at Leicester University where I completed my PhD in immuno-oncology research with Professor Martin Dyer, and I have held previous roles at Cancer Research UK and ABCAM. So the key take-home messages today are really that clinical and non-clinical data for BiTE and CAR-T therapies have highlighted the significant possibilities to optimise both BiTE and CAR-T. VHH can be used to refine tumour cell targeting and modes of action for bispecific antibody T-cell engagers and CAR-T with carefully defined affinities, valencies and specificities. So a quick introduction to Isagenica. Isagenica have been specialising in VHH single-domain biotherapeutics discovery for many years. We are based on the Chesterford Research Park in Cambridge, UK, and our team have over 20 years of experience in therapeutic antibody discovery with backgrounds in leading pharma and biotech. We have a consistent track record of delivering clinical assets through our collaborative partnerships. Isagenica have Llama VHH generated from our proprietary state-of-the-art, highly diverse synthetic VHH library, constructed with patented Calibra design. Combined with our own cis-display technology, our library interrogates more than 10 to the 13 library members, offering optimal expression of maximized functional diversity. We developed Llama VHH to construct next-generation biotherapeutics for the treatment of cancer, inflammation, and other serious diseases. VHH are small in size, biophysically robust, and readily manufactured, and have notable attributes making them highly versatile small-format antibodies, lending them to a variety of therapeutic avenues. We will be touching on many of these features as we progress through the rest of this talk. VHH single domains are one of the most accepted next-generation antibody therapeutic formats. Caplacizumab, or Cablivi, is the first FDA-approved single-domain antibody therapeutic. As you can see from the table, VHH antibodies are being developed in early clinical trials through to late-phase studies encompassing a range of disease areas such as autoimmune and heme and solid malignancies. So far, VHH are being developed in a number of formats, including monomeric, bispecific, half-life extended, and CAR-T formats, highlighting the versatility of this type of antibody as a next-generation biotherapeutic. As shown in the previous slide, not only are VHH versatile, they are easily formatable, including into antibody-drug conjugates and direct therapeutics, particularly in the case of challenging targets where their small size offers an advantage for binding. The focus of today's talk is a more detailed view on the application of VHH as components in T-cell redirection therapies, and how their incorporation can enhance the therapeutic window, tune target affinity binding, and enhance biomarker specificity to minimize off-target toxicity. As a precursor to this, I will now hand over to Dr. Ed McGowan, who will describe the features and attributes of VHH in more detail.
Many thanks for that introduction, Mandeep. So I'm going to be talking a little bit more about the VHH structure and function and how those might be applied to T-cell immunotherapies. In the early 1990s, researchers discovered that camels and llamas produce heavy chain-only antibodies. As a consequence of this, we can isolate the 15 kilodalton VHH from those heavy chain-only antibodies, in contrast to the IgG of a human, which typically displays the heavy and light chain, generating the more complex tertiary folded Fab structure. The small size of the VHH is approximately the 10th the size of an IgG molecule, and its simple structure opens avenues for multiple different formatting options. Some next-generation biotherapeutics utilize VHH and other single-domain antibodies into a range of diverse formats. So VHH-only molecules might be bispecific, which is shown in the top left panel, bivalent, which is shown by the dark and the grey panel, or multivalent. And Isagenica is working up to tetravalent and more in these kinds of constructs. We can also utilize VHH to modify the antibody-drug conjugates or peptide conjugates, giving them features such as half-life extension or redirection. And that's something that we can also translate onto more traditional IgG-based therapeutics, be those human or mouse in origin, where we can add the IgG, adding a secondary function to the antibody. And that could be by modification of the Fc, the light chain, the Fab domain, or in fact any of the above, or a mixture of those things. And those kinds of, all of these kinds of molecules are currently in different phases of clinical development. The VHH makes excellent building blocks for non-IgG therapeutics. I've already mentioned their size of 15 kilodaltons. That translates to about 3 nanometers squared in area or volume. And that's of particular relevance in the context of the immune synapse that we'll be discussing later for T-cell redirection therapies, which has to be in the order of 30 nanometers. The natural occurring structure means they've evolved to be very stable. It also offers some really nice engineering options because they're considerably simpler to modify as a consequence of them having no quaternary structure or quaternary-like structure that would be associated with an scFv. They have a very, very good immunogenicity profile, and that's a consequence of them being small. They have a very high homology to the human VH, and because of their single-domain origin, they have an absence of any exposed hydrophobic regions, all of which make them very poor immunogenic targets for the immune system. And that means that their use as a therapy is potentially improved as a consequence of the unlikely generation of antibody-drug antibodies. Isagenica's synthetic VHH library has been developed with a number of different features that make the leads identified from it easier to develop. First of all, it's a very quick process. We don't have to immunize with our process, and that means that we gain a 2 to 3 month advantage over typical immunization protocols utilized with whole llamas. We can use all types of selection antigens, and Isagenica has experience of identifying VHH from our libraries against GPCRs, ion channels, soluble targets, and intercellular targets. And we use a multiple range of things, whole cells, purified protein, or VLPs to enable that. The quality of the hits that we generate from our libraries are quite remarkable. We have high diversity and we have high affinity on a first pass, and I'll show just a little bit of exemplar data of that in a moment. The other advantage of working with a synthetic library is obviously the increased epitope and sequence diversity. We aren't working towards immune dominance, we're working towards a broad range of epitopes and essentially optimizing and selecting the epitopes of interest from our synthetic approach. As I mentioned, we developed in some nice features, and one of those was the lack of liability. So the library is being developed with an optimized sequence, enabling a clean progression into a CMC program and therapeutic exploitation. We've also humanized the molecules to give an even improved immunogenic profile, and I'll show some data about that in a moment too. Here are some typical VHH biophysical characteristics for molecules identified from our library. You can see that from the SEC data on the bottom panel that we have discrete peaks showing a nice monomeric VHH molecule. And from a stability perspective, in this example, we're showing about 60 degrees melting temperature, which is very good for without any kind of maturation from the library. And typically, the range is in the order of 60 to 70 degrees for our lead molecules. As I mentioned, we've also engineered in humanized frameworks to reduce immunogenicity risk with our libraries. And this is an example of some data generated by immune experts who assessed the typical VHH sequence from our libraries for linear T-cell epitopes. This is done computationally, scanning the entire molecule for dominant HLA binding motifs from real-world data sets. And what we see here is the top panel, which represents some clinically approved antibodies, and the bottom panel, which represents either a camelid or a humanized variant from our own VHH synthetic libraries. The panels are broken down into the world average or regional HLA dominance, because HLA dominance varies across different populations. And we've highlighted the EU, US, and Japan, which are the major regulatory bodies typically associated with therapeutic development. What you can see in the top panel is that whole antibodies typically return a score in the order of a 6 to about 4.5. And these are arbitrary numbers, larger numbers indicating a greater provenance of T-cell epitopes. And on the bottom panel, you can see that at the same kind of breakdown, our camelid versions, without any kind of humanization, are returning numbers that are broadly speaking in keeping with the immunogenicity profile of an IgG-type molecule. But the humanized variant, you see, has an immunogenicity profile that is even improved upon any of these IgG-based therapeutics. So again, we have a small size, solubility, and stability that makes the VHH a very poor immunogen. But we've also engineered in further humanized sequences, which makes the immunogenicity profile even better in the context of utilization in subsequent therapeutic avenues. It's very important to be able to format the molecules that we're working with, particularly in the context of T-cell engagers, making multivalent binders. And this is just some data that we generated again from our libraries, where we identified VHH, a number of VHH against IgG, and that's shown in the left panel. And so we have VHH against the Fc, the constant heavy chain, and then the constant light and the variable light chain. And those produce EC50s in the order of 1 to 5 nanomolar. In the middle panel, we looked at what would happen if we made any of these into a bivalent, and here we're showing the Fc domain, which has been made into a bivalent molecule through a linker region. And you see that the monomer has approximately 5.5 nanomolar EC50 binding to the IgG. When we make it bivalent, we get a 16-fold improvement, dropping that to 340 picomolar, which is really quite remarkable data. When we look at avidity to adjacent epitopes, or bispecific molecules, then what you see here is the individual VHH are returning about 1 to 5 nanomolar potency. When we put them together into a single entity, we get a 50-fold enrichment in potency, dropping to about 100 to 120 picomolar. And that shows how versatile the VHH are when it comes to formatting in this very simple model system. So in summary, VHH are an attractive option for T-cell engagers. They're a stable single domain of small size, which can be generated from immune and synthetic libraries, and Isagenica has generated very advanced and validated synthetic approaches. We have humanized constructs, which improve their developability, and the VHH in itself has been clinically validated. We can tune the affinity, we can tune the formatting, we can introduce variable linkers to generate avid binding or multivalent targeting. And that's a consequence of the flexibility the VHH display with respect to steric geometry. I'll now hand back to Mandeep, who's going to discuss some of the clinical landscape associated with T-cell immunotherapies.
Thank you, Ed, for that little summary. So, a quick history of immunotherapies and the clinical trial landscape. Immunotherapy for cancer has been used in the clinic since the 90s with the introduction of rituximab for the treatment of non-Hodgkin lymphoma. Since then, antibodies have been used extensively in the treatment of cancer, with over 60 antibody-based drugs approved for therapeutic use and currently marketed, with worldwide revenues of $98 billion in 2018. The timeline shown is an adaptation from a thorough review of both CAR-Ts and BiTEs by Nassau and Stroll, and would highly recommend you read as it elaborates on the themes covered today. As an idea, T-cell immunotherapy has been around since the 1980s, demonstrated using autologous tumour-infiltrating lymphocytes and the use of bispecific antibodies killing tumour cells in vitro. So far, only 2 autologous CAR-T therapies have been approved, both in 2017. These are Yescarta by Kite Pharma and Kymriah by Novartis. Both have achieved extraordinary results in B-cell malignancies, targeting CD19 with a single infusion of engineered T-cells, achieving remission rates of up to 94% in patients previously found to be unresponsive to standard treatments. Allogenic therapy, which relies on a single source of cells to treat many patients, has no approval so far. Blincyto, which is the BiTE developed by Amgen, gained approval in 2015 and has been most extensively used to treat acute lymphoblastic leukemia. It is the most advanced CD3, CD19 targeted immunotherapy. Survival data shows that more than half of patients were alive 5 years following the first cycle of treatment. Compared with autologous cell therapies, BiTEs offer an off-the-shelf immunotherapy solution, making T-cell treatment accessible and affordable to healthcare providers and patients. The remarkable efficacy of CAR-T has generated significant investment in these treatments, receiving $12.8 billion in 2018 alone, demonstrated by the high proportion of CAR-T in early phase development. The majority of allogenic therapies are in phase 1 development and comprise only 10% of their autologous CAR-T equivalent, showing there's much progress to be made before this will be accessible to patients. Interestingly, more than half of the clinical trials for bispecific antibodies target CD3, the majority of which are in phase 1 clinical development. The top 5 indications for both CAR-T and CD3 T-cell engagers are being developed in target hematological B-cell malignancies, where success rates are expected to be higher than for solid tumours. This points clearly to the continued development and improvement of these therapies. Despite positive clinical results, both therapies face similar safety concerns, including cytokine release syndrome and neurotoxicity. CAR-T and antibody-based T-cell engager therapies are in development for solid tumours. However, to date, there are no approved therapies for treatment. Current BiTEs require continuous and extended intravenous infusion due to short half-life and narrow therapeutic window. High-affinity targeting to CD3 can also cause accumulation of the bispecific T-cell engagers in T-cell-rich tissues rather than the intended tumour environment, leading to unwanted off-target activation of T-cell, worsening toxicity and reducing therapy efficacy. There also remains complex operational barriers to wide-scale adoption of autologous CAR-T therapies, and this includes the need for a certified centres for administration, multi-week manufacturing process, and extended duration of patient hospitalisation. Allogenic CAR-T therapies are being developed to create an off-the-shelf standardized approach to overcome these hurdles. Such a therapy would, in theory, allow for redosing or combination of CAR-T cells directed against different targets and decreased cost using an industrialized process. The contrasting mechanistic features of bispecific T-cell engagers and CAR-Ts creates differing opportunities and challenges to optimize their safety and efficacy. I will now hand over to Ed, who will further elaborate on how VHH can be used to refine tumour cell targeting and modes of action for bispecific antibody T-cell engagers and CAR-T, with carefully designed affinities, valencies, and specificities.
Thanks again, Mandeep, for that introduction to the next phase of the project. So I'll be looking at some case studies exemplifying VHH and their advantages in CAR-T and T-cell engagers. So here's just a little bit about the CAR-T and T-cell engaging mechanism of action, which are very similar but just slightly different. So in the context of the CAR-T, the T-cells of the patient have to be extracted and transformed with the chimeric antigen receptor. This then when those T-cells are redeposited in the patient, those get redirected to the tumour via that chimeric antigen specific receptor, and upon engagement with the tumour cell, you get a specific proliferation of those CAR-Ts, which is HLA independent. The story for the T-cell engagers is slightly more complicated. Here, soluble T-cell engagers, in this case, there's an example given of blinatumomab, the Amgen BiTE molecule, are recruited to the tumour cell, again, by a tumour-associated antigen-specific entity, in this case, CD19. And then that accumulation of the BiTE molecule recruits T-cells non-specifically via CD3, non-specifically in the context of HLA independence. Again, just mentioning here something that I mentioned earlier, the immune synapse needs to be approximately 30 nanometer in diameter for efficacy, and the VHH has a diameter of about 3 nanometers, meaning that we have a great advantage there. Engagement with a tumour cell by the T-cell as a consequence of the BiTE leads again to tumour cell killing, T-cell activation, and T-cell proliferation. The activation and the mechanism of action of both these entities, the CAR-T and the T-cell engagers, and the non-specific nature of that means that cytokine release syndrome is a very recognized and known complication associated with these therapies. And in extreme cases, that can also extend to neurotoxicity profiles as well. And we need to do a lot of work within the space to be able to improve upon those. And I'll be talking about that a little bit later. CAR-T are remarkably efficacious, but what does that really mean? Here, Schultz et al. Compared the real-world data to look at and compare CAR-T therapy to hematopoietic stem cell transplantation or bone marrow transplantation. And they looked at the 3-year probability of relapse-free survival. And they found that for CAR-T, this was only 46%, whilst for HCT, it was 68%. Now, there are a couple of caveats here. Clearly, a more severe disease burden associated with CAR-T treatment at the moment, and also the data sets are based upon the first generation of therapies that are currently approved. But what this shows is that next generation therapies are certainly warranted in this field. Now, why does this relapse occur? There are 3 main mechanisms for the therapeutic escape of the tumour as a consequence of T-cell redirected therapy. The first is antigen escape. Here, epitope mutation gives a competitive advantage to a particular tumour following therapeutic pressure, and that then expands to become a dominant cancer strain. Or you could have lineage switch, and this is particularly relevant in the context of B-cell lymphomas, where a malignant line modifies its phenotype under therapeutic pressure to become the new dominant strain. And finally, there's antigen downregulation. This would be relevant in both the hematological and solid tumour fields, and is similar to antigen escape, where essentially the target is downregulated on the surface of the cell, rendering the redirection therapy ineffective. How can we overcome this kind of issue with our therapeutic targeting? Here are some examples given for the CAR-T field, and some of them are relevant for both CAR-T and bispecific targeting. So the first is to make a bicistronic CAR. And this is a more complex CAR insert and has potential promoter challenges associated with it. VHH would give an advantage here because the simpler inserts would lead to improved expression. And of course, the VHH being a single domain antibody with no quaternary structure wouldn't suffer from the scFv cross-pairing issues that are seen when multiple scFv are in close proximity. The tandem CAR-T has the highest similarity to that of the bispecific T-cell recruitment model, and in here, a VHH, 2 or more VHH would be inserted in a single CAR and lead to a more stable presentation on the surface, again, as a consequence of no cross-linking. Co-administration could also be administered within the CAR field, and this may be a good clinical answer with likely sequential administration of different CAR-T therapies, but it might be difficult to obtain enough cells for a generation of this in an autologous fashion. And then finally, there's co-transduction. This would lead to a complex heterogeneous product, and that might be quite difficult to control from a CMC batch production approach as the CAR-T are being generated. So I mentioned a couple of times on that last slide about the CAR-T manufacturing process. And this is quite a complex and difficult process to get right. And it's broken up here into 6 stages on the left panel. Firstly, the patient has been through multiple chemotherapy by the time this phase of treatment has occurred. And that could lead to a compromised immune system, meaning that the first stage of leukapheresis could be quite a challenging process. Once the T cells are generated from the leukapheresis, they are enriched and activated. Those activated cells are then transduced with a lentiviral CAR construct and then further expanded to generate an expanded panel of CAR-T expressing T cells, which are then further isolated to produce the final product. While stages 2 to 5 are being performed, the patient is undergoing lymphodepleting chemotherapy. Again, a quite dramatic procedure for the patient, which is required to give a niche to enable re-establishment of CAR-T therapy on the administration of CAR-Ts at phase 6. Clearly, this is an expensive and challenging process, and it makes CAR-T therapy a late-stage treatment. The generational CAR-T improvements that some people may be aware of is largely focused to the signaling, which has improved the persistence and cytotoxicity profiles of CAR-T and also their safety profiles. There's 4 generations of those, and they're typically based upon the signaling profile. But it shows that we still have more to do on the targeting approaches. Bispecific T-cell engager fundamental features are more based upon the affinity associated with the two binding arms. And Dr. Safar from Amgen produced a very nice study that was published in 2017. So all bispecific molecules for T-cell redirection need to bind to the T-cell and the tumor. And the accepted dogma is that higher affinity tumor, lower affinity T-cell binding arm would lead to accumulation of the therapy at the tumor and localized activation. And associated with that would be an improved safety profile with less off-target activation and a potentially improved cytokine release syndrome type profile too. Here, that was directly assessed by looking at 3 different variants of the T-cell engager. So it's a high, medium, and low affinity variant, and also a high, medium, and low affinity variant of the tumor targeting agent, in this case, an anti-CD38 mAb. In panel C, we see that the 1, 3, 6, and 8 have been applied. Now, in the data that this generates, these are based on a CD38 high and a CD3 high for panel 1. Molecule 3 is a CD38 high and a CD3 low affinity molecule. Molecule 6 is a CD38 low and a CD3 high affinity molecule. And molecule 8 is a CD3 low and a CD38 low affinity molecule. And what's most striking here is that all of the high, low, intermediate molecules, 1, 3, and 6, reach the same level of saturation, where molecule 8 doesn't return any cytotoxicity at all. So that's really good to know that just using low affinity molecules isn't enough to generate T-cell activation, but any combination of high and low affinity molecules will generate activation, which saturates cytotoxicity with 100% for all of those molecules, though the potency of those is slightly different. Panel D looks at this in slightly more detail and looks at the high, low, and high, medium affinity interactions. And what you see here is that Amgen decided that the medium affinity interaction was the most likely to give the desired effect required. And that's a consequence of the data shown in the top right panel, in panels B and C, where we look at the impact on cytokine release associated with these molecules. So if we have a high or a medium or a low, you see that the high generates consistently more interferon gamma and TNF than either the medium or the low. Now the low doesn't produce any cytotoxicity, so the medium affinity molecules, and those are molecules of around about 10 to 20 nanomolar for both CD3 and the tumor-associated antigen, give 100% cytotoxicity with a reasonable potency, but a reduced cytokine proliferation event. When we look at how these actually responded in the animal model here, which was a bone marrow invasive cancer model, you see that at day 7, the vehicle and AMG 424 are both looking very similar in their bioluminescence profiles. However, by day 18, the mice who received the vehicle are completely overrun by the cancer and have significantly brighter bioluminescence relative to the animals that received IMG 424, which essentially have no bioluminescence present at all. So this is a really nice study looking at the relative affinities required to generate potency and likely safety associated with a T-cell engager. VHH in bivalent redirection have some inherent advantages, and I've mentioned these a couple of times throughout the talk. They're able to bind avidly, giving gain of selectivity or prevention of escape. But in generation of these bivalent molecules, we have advantages over the SCFV constructs that many organizations use. And that's because when two SCFV molecules are in close proximity, as they would be in a BiTE, there's a propensity for the VH and the VL of each SCFV variant to cross-link with each other. And obviously, cross-linking would completely abrogate the binding of the paratope of that particular SCFV and render that therapeutic effectively ineffective. The single domain nature of the VHH with no quaternary-like function completely abrogates this kind of risk, and so would lead to an improved manufacturing efficiency of the manufacturing process associated with CAR-T, or in the context of T-cell engagers, improve stability and potentially flexibility upon application. So looking at CD19-20 tandem CAR-Ts in diffuse large cell B-cell lymphoma, Tong put this really nice paper together that describes some of the features of these CAR. In the top left panel, we have features A and D, which show a remarkable survival rate for tandem targeting. In A, we have the non-transfected model. We have the CD19 CAR showing good survival at day 21, and the CD20 CAR, which shows that there's the still presence of the tumour at day 21. Looking at the tandem CAR, we see that the mice have completely cleared the tumour. And that's shown in panel B, which quantifies the bioluminescence and also addresses in panel C the improved T-cell trafficking. Panel D shows the remarkable survival rates, and you can see that the tandem CAR has its first death after over 40 days, whereas the CD20 CAR, the orange line, shows that we have all of the mice subjects have unfortunately died before that time point. And panel E is showing that the tandem CAR are at termination clearance, because of the clearance of the tumour, you have a considerably reduced CAR proliferation within those zones. That's then being translated over to the clinical features, which shows remarkable efficacy. Here we have the objective response rates and the partial response rates and the complete response rates. And in panels D and C, we can see that at 2 to 3 years, we have 90% survival. And at that 90% survival rate, 75% of those are with complete remittance, which is really remarkable when you look at the context of the first generation CAR products, which were compared earlier, which were shown to have a 46% remission-free survival rate at 3 years. So bispecific tandem targeting of CAR-T is going to be a really powerful mechanism as we move forward in this field. And as I've shown, the VHH would be considerably more efficient within this context. Here we're looking at bispecific T-cell engagement, and more specifically, bivalent targeting of CD20, and utilising the glofitamab Roche TCB molecule as an example of this, which is currently showing excellent clinical efficacy in trials. In the example given here, tumour cell lysis was assessed utilising a PBMC co-culture model with 3 different cell lines. Clearly, the bivalent targeting of the 2-to-1 CD20 TCB in panel A, as shown as the red line, is showing greater efficacy and potency than either of the monovalent arms of the CD20 TCB or the IgG CD20 TCB. However, we think it's really quite important to point out that the CD20 TCB 1-to-1 OA, shown with the black line, is more efficacious than the 1-to-1 CD20 TCB in all 3 of these co-culture PBMC killing assays. And that's likely a consequence of the tighter synaptic bridging that's occurring, leading to a more efficient formation of the immune synapse. Panel B looks at the impact of the FC on the activation, and no change was observed, meaning that the features associated with the improved efficacy are purely a consequence of the presentation of the CD20 bivalent molecule. Solid tumours offer a completely different range of challenges to those faced for T-cell immunotherapy engagement in a hematological oncology setting. And the reason for this is largely a consequence of the tumour microenvironment, or stroma. Tumours are self, and the environment has evolved to downregulate potential autoreactive immune responses in the healthy tissue. And so to overcome this, we need to look at how we might get by this. And again, targeting is the key. Bivalent targeting to highly expressed TAA in, for example, HER2 in breast cancer, might effectively redirect the T-cell therapy away from low-level expression of the same targets in this example would be cardiac tissue. Another option would be bispecific tumour targeting. For instance, in melanoma, where CD70 and B7 have both been identified as highly robust biomarkers, The use of both of these in a T-cell redirecting therapy would ensure that the T-cell redirection was localized only to the melanoma and not to off-target approaches. And that would be relevant in both cases to both the CAR-T and soluble T-cell redirecting field. Another option that we aren't going to be discussing here because it's not quite so relevant is the co-administration of checkpoint inhibitors to enable the recruited T cells to retain activity rather than be downregulated by that stroma microenvironment. Here's an example of VHH CAR-T used in solid tumours. So this is again a mouse model, and it's a mouse model to a colon cancer model. And it was performed by Joyzin using the VHH to PD-L1, the model of this being to recruit the T cells to areas where PD-L1 was upregulated. Again, that would be in the tumor stroma environment. What we can see on the top panel is the dosing regime. But interestingly, straight away, jumping into the data, we see that when we look at VHH CAR-T, we see complete remission of complete reduction in the tumor area. Whereas where vehicle is being provided, then the tumor grows exponentially. When we look at the infiltration of the cells into that solid tumor, we do see that CD3, 4, and 8 are all upregulated in the tumor infiltrate. And when we look at what those cells, the live dead features of those cells, we see that in all cases, the CD3, 4, and 8 are functional and alive. So clearly, VHH can be used for CAR-T treatments in this very interesting and novel application of targeting of a checkpoint inhibitor. The soluble VHH has some significant solid tumor advantages, but this is a dogma that's been around but rarely addressed. Here, a study was performed that looked at the correlation of the affinity and size on the infiltration of VHH into the tumor in comparison to an approved antibody therapeutic. So in the panel to the left, we compare the affinities of a monovalent, a bivalent, a bispecific, and an antibody. And we see that the monovalent molecule, panel A, has relatively low affinity for the target. When we look at panel B, we see that the bispecific variant retains that low affinity, that's comparable to the monovalent, but is obviously doubled in size to approximately 30 kilodalton. When we look at panel C, on the left, we see that a bivalent variant of monovalent A has high affinity and, again, doubles the size of the molecule. And when we look at the antibody molecule, which is approximately 10 times the size of the monovalent VHH, we have both high affinity and size. Looking at the panel to the right, we see the established tumours at 1 and 24 hours post administration of the different VHH or antibody constructs. And most interestingly, if you look at the first hour at the top right, none of the antibody shown on the far right panel of the different mouse bioluminescence whole body images has infiltrated into the tumour. Whilst at 24 hours, the antibody is found to be enriched only in the kidneys and bladder. Looking at the mice that have received the VHH, the pattern is showing in all cases accumulation in the tumours. If you look at the bottom panel, this quantifies the bioluminescence, and I've highlighted two panels in purple and yellow, which compare the affinity and size respectively. Looking at the two purple panels, we see the bispecific lower affinity molecule shows greater bioluminescence over time relative to the bivalent high affinity molecule, showing that a lower affinity bivalent VHH is able to infiltrate the tumour more effectively than a high affinity bivalent VHH. Comparing the affinity, the size of the molecules, we see that the high affinity bivalent VHH, which is relatively comparable in affinity to the antibody molecule, does show infiltration, whereas the antibody molecule, which is 5 times larger than the bivalent high affinity VHH, essentially shows no tumor infiltration at all. So this is a really nice example that highlights the utility of VHH in solid tumours in a soluble format. Solid tumours and VHH T-cell engager redirection is also important in the context of the orientation, which can have dramatic impacts in the efficacy of the resulting bispecific molecule. Here, VHH to EGFR and gamma delta 2, a marker of gamma delta T cells, was utilized in the treatment of a colon carcinoma model in mice. Looking at panel A in the middle, we see that the orientation of the VHH has a large impact on the VHH binding to its targets. In panel B, we see that in this case, the length of the linker region has a very limited impact. Again, I'm going to remind us that Isolink have generated our own platform to assess this kind of impact with the Isolink platform. On the right-hand side, we then see the efficacy within the colon cancer model, and we have the schedule of the work done in panel A with induction of the tumour at day 0, followed by treatment at days 1, 4, and 7. In panel B, we have the bioluminescent whole body imaging scans, and we see that when PBS is used in the top panel, the mice are completely overrun by the colon model at 35 days. When a control antibody is used, the mice are again largely overrun by the colon carcinoma cell line. Gamma delta T cells, which in themselves have some quite remarkable features and with respect to potential treatment of tumour therapy, have shown some remittance of the tumour in the third panel, whereas when we mix the gamma delta T cells and the T cell redirecting therapy, the mice have complete ablation of the tumour. And when we look at panel D, again, we see the remarkable survival curve associated with a bivalent or bispecific T cell redirection program, with the first death occurring after all animals have died in both the PBS and antibody control models here. Bispecific potency is heavily impacted by half-life extension. Small molecules less than 30 kilodaltons have a short half-life of less than 30 minutes as a consequence of filtering by the kidneys. Utilizing binding of the VHH to albumin, we can extend this PK profile to a half-life in the order of 4 to 7 days, utilizing VHH at different affinities to tailor that half-life. This means that the treatment regime can go from the current standard of an infusion approach to repeated, less frequent dosing, and even potentially transfer the therapy to an outpatient profile. This has significant improvements in the therapeutic window and patient experience associated with these kinds of molecules. So in summary, VHH offer numerous advantages for both CAR-T and by therapeutic development. The simpler structure of the VHH infers a simpler manufacturing process in both cases, and that can be transcribed into a lower cost of goods. The VHH are stable and have a very low immunogenicity profile, meaning long-term dosing regimens can be applied within the patient population. Making multi-specific variants, they show no cross-pairing issues that might be associated with the scFv, and we can tailor their half-life in the context of soluble by T-cell engagers to improve the dosing regimen. That major advantage is transferred into the CAR-T field when we look at the bicistronic or the tandem approaches, which in both CAR-T and soluble engagers has shown remarkable efficacy improvements in both models and the clinic. I'll now hand over to my colleague Mandeep, who will conclude.
Thank you, Ed, and thank you for a thorough review of the application of VHH in both CAR-T and bispecific formats. What Ed has demonstrated is that there is a clear scope to further optimise bispecific antibody T-cell engagers and CAR-T using VHH, highlighted by some interesting and elegant studies. This raises the hope of curative therapies for a wide range of malignancies. Both formats show much promise, however, progress hinges on resolving their shortcomings of toxicity, tumour escape, and extending their application to the treatment of solid tumours. Bringing these clinically proven technologies together offers a highly promising route to next-generation T-cell redirecting therapies that more effectively harness the immune system to fight cancer. Many of the topics covered today are summarised in our white paper, which can be downloaded from our website. You can also follow us on LinkedIn to follow any developments and progress of Isagenica in this field. Also, if you'd like to have a further discussion about any of the topics we've covered today or the application of VHH to your own discovery projects, please do contact the Isagenica Business Development team. Thank you for your attention, and we'll now move on to Q&A.
Thank you very much, Mandeep and Ed, for that presentation. It is now time for the question and answer session. To ask a question, please type it in where it says type your questions here and then press submit. So to get us started, I'd like to ask you if any partnered VHH assets have reached the clinic yet. Mandeep?
Yes, thanks for that question, Sarah. So Isagenica have run partnered programs where VHH have been applied in various formats. We have 2 partnered assets currently in phase 1 and phase 2 development. However, because of our confidentiality agreements with those partners, we're not able to disclose any further details about them.
Okay, thank you very much. Our next question asks, are VHH a better format of antibody than SCFV?
I think I should probably take that. Yeah, we would argue yes, and that's hopefully being displayed throughout the talk. Basically, their small size and their very stable format makes them just much easier to engineer and work with when you're going through that discovery development process. They're very easy to engineer, and they have a very good immunogenicity profile, and that's really useful, again, in that development process. We don't want to be generating anti-drug antibodies, and so they're just very, very good for that. Obviously, SCFV are approved, and SCFV are used in quite a lot of different contexts, but that's probably more a consequence of just their ubiquitousness. We do think that the use of VHH at the front end of a project would improve the efficiency and the developability of the drugs that come out of those discovery programs. So yes is the short answer.
Thank you very much. A quick reminder to our live audience, you still have time to ask a question. To do so, please type it in where it says type your questions here and then press submit. Our next question then, do Isagenica have internal programs or data that would demonstrate the application of VHH in either format?
We've got a number of internal programs running at the moment. We have partnered and internal programs. So, and within those programs, we tend to work towards a formatted structure. The nature of VHH lend themselves to that, and it's a major advantage in doing so. So yes, we do have a number of programs running right now. Again, for the same reasons as Mandeep mentioned earlier, we can't go into the details of those due to confidentiality.
Okay, thank you very much. Our next question. What affinity have you reached in maturing your VHHs? Is there a limit in achieving PM affinity because of a single domain?
We've taken our clones to sub-nanomolar affinity through affinity maturation. We typically get a range of affinities from kind of double-digit nanomolar down to single-digit nanomolar straight out of the libraries that we investigate, and I showed a little bit of data alluding to that earlier. But with affinity maturation processes, we can take those down to sub-nanomolar in the picomolar range. There's no the limits affecting the VHH are comparable to the limits affecting any kind of antibody or scaffold antibody like binding fragment. So there's no kind of differentiator there between a VHH or another format, except for, again, going back to that ease of formatting. Because we're a tertiary structure and not a quaternary structure, it's really quite straightforward to run these kinds of programs because we can quite easily predict impacts on the stability of the molecule as we go through the affinity maturation program. So yeah, I hope that answers that question.
Thank you very much. Our next question asks whether there have been any studies to show that VHH are more efficient at forming the immune synapse compared to the alternative.
I haven't seen head-to-head studies like that. We are performing some work that's related to this in-house at the moment, but won't go into too much detail there. It's still in development. But for the reasons that we said, again, going down to the size, the immune synapse is very, the efficiency of immune synapse formation is directly proportional to the distance between the T cell and the target cell, and the VHH is approximately a quarter the size of, a VHH is approximately half the size of an SCFV. And so a bivalent or a tetravalent VHH is still going to be smaller than a typical SCFV molecule. So we would predict that the use of VHH in that kind of format would be advantageous for an efficient synapse formation. And obviously, we did demonstrate data today that showed that VHH have been used in bispecific engagement type programs with very efficacious outputs. So, yeah.
Great, thank you very much. Our next question asks, what is the developability assessment if you combine two VHH in constant, heavy and light chain in regular monoclonal antibody format?
I'm sorry, could you just repeat that question? It didn't come through very well.
Here it is again. What is the developability assessment if you combine two VHH in constant, heavy and light chain in regular monoclonal antibody format?
So if you were to use a VHH, I think I understand that. If you were to use a VHH in a regular monoclonal antibody format, it would look quite different actually. So because you don't have a light chain, it's a heavy chain only antibody fragment that it's isolated. The VHH is isolated from. Your antibody would have an FC with the two VHH hanging off the ends where you would typically have the Fab fragment. So I think the developability assessment, it would be quite straightforward. It's quite common to use an FC as a stabilizing agent or a formatting tag or entity for these kinds of products. So essentially, the developability assessment would be to put the VHH on the end of the FC through molecular formatting, so it would be produced that way, and with a flexible linker likely, and then we would be assessing for subsequent binding, retention of binding capacity relative to the source VHH that was used in the generation of that construct. Yeah, it's a good question. Thanks, Lee.
Great, thank you very much. Our next question asks, have you developed a VHH specific for CD3, and is it better than current anti-CD3 SCFV?
We couldn't really talk about the specifics of our programs, unfortunately, but we are very interested in immunotherapies and working towards some of those kinds of goals that were described in today's presentation.
Okay, thank you very much. A reminder to our live audience, you do still have time to ask a question. To do so, please type it in where it says type your questions here and then press submit. Our next question then, what is the business model used by Isageneca when working with its partners?
So Isageneca like to work as partners, so we like to take time to understand projects and also sharing the successes of our partners as well. We usually develop a work plan according to a target product profile, which is really the aspects required for the therapy and the target in mind, and those are based on the partner requirements. So we're always open to having informal chats about projects, so please do contact BD@Isageneca for further information.
Okay, thank you very much for that, Mandeep. Our next question asks, what is Isolink?
Yeah, I did mention Isolink. So, yeah, that's the formatting of VHH or any kind of bivalent entity takes quite a lot of empirical optimization, and to accelerate that process, we developed Isolink, which is a program of work that or a panel of linkers that have defined structures. So we have flexible, semi-structured and fully structured linkers with also contain variation in length of the linker region. So it provides us with approximately 30 different constructs that enable us to quite efficiently and quickly assess the steric implications of formatting and to be able to advance molecules in bivalent or multivalent entities far more quickly than we'd have to do that in a kind of unguided fashion. So yeah, I mentioned that on one of the slides earlier. I'm glad that somebody clarified that question. Thank you.
Thank you very much for that clarification. Our next question is on the immunogenicity of VHHs. Does the humanization approach compromise the biophysical chemical properties of the VHH? Is it vital that it's done? Can a fully human VH be immunogenic in humans? I'm not sure I understand the last section, but
no, that's okay. Thank you. No, that's a good question. So the VHH are very similar in sequence, straight out of the bag, straight out of the box, with respect to similarity to the VH sequence of the SCFV. But what we have is the ability to identify key residues, and those are very well described, that lead to an improved humanization profile. And in the humanization of a VHH, we would be assessing the biophysical impact as we go. So we'd be making, first of all, we'd be making random specific mutants, and then we'd be assessing the impact on biophysical properties, and then we'd be making combinatorial libraries based on those. So there's a bit of a process involved in that humanization, but we always assess the impact on the biophysical outcome as we develop those. And it's quite empirical and related to specific CDR interactions with the frameworks. And so to get around that, we've actually developed humanized libraries. So because we work with synthetic libraries, the backbone that we work off is already humanized, and so the CDRs are presented in the context of a humanized backbone, and that means that the developability of the leads that we generate, in essence, are already optimized for a humanized format. So it's already lowly immunogenic, and that humanization process is something that we don't really need to worry about too much within Isageneca. Could it be immunogenic in humans? If you try hard enough, any antigen will become, or any construct could become immunogenic, but the immunogenic profile of the VHH, as I showed, is very good. They're very small, so that tends to make them a poor immunogen. They're hydrophilic, so they tend not to display unwanted residues that are prevalent in typical kind of antibody or T-cell mediated epitopes. And when we've done the in silico epitope scanning of sequences from our libraries, we find that they are improved upon those of typical human IgG therapies, and the humanized variants of our library are again improved over the camelid variant of our library as well. So we think that we've done a very good job of de-risking our VHH platform in the context of immunogenicity, but that is a very important thing to consider as you're going down a antibody development route, because obviously the end therapy needs to be potentially dosed over long periods of time, and that could be an issue. So thank you. Hopefully I've answered that question.
Thank you very much. Our next question asks how we can use VHH for ELISA, IHC, or IFA.
How do you use them for ELISA, IHC, or IFA? So they would be able to be used interchangeably with, as you would, a typical antibody. We actually don't generate tool antibodies very regularly here at Isageneca, but they have a similar kind of antibody specificity as you would associate with any antibody fragment or scaffold or antibody. So they could be labelled directly with fluorophores or with enzymatic type labels for use in ELISA, and you would be able to use them accordingly. They may have some advantages with things like IHC and IFA with respect to tissue penetration and so on. So it would be quite straightforward, either through a biotinylation process or a direct labelling. Yeah, I don't see a problem with using them for that format if necessary. Yeah.
Okay, thank you very much. And that is where we need to finish for today. We are aware that there are a few questions that we didn't manage to get to, and we will get back to you offline after this broadcast finishes using the details that you've provided. I'd like to thank Dr. Ed McGowan and Dr. Mandeep Semi once again for their presentation and for answering your questions. I would also like to thank the webcast sponsor, Isageneca, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com/webcast. Thanks for watching, and I hope you'll join us again soon.